Hi students, today we will discuss about vitamins and before that enzymes and vitamins both we will discuss today in this session. So please kindly subscribe my channel. So enzymes means what? Standard 12th, this is chapter standard 12th biomolecules only, chapter name enzymes. So what is enzymes? What is enzyme? Enzymes means it is also like proteins. It is also like proteins. Enzymes are defined as enzymes are defined as biocatalysts produced by, it is a catalyst produced by living organism or living cells. It is produced by living cells which are catalyze many biochemical reactions, not chemical, biochemical reactions. So this means what? Enzymes are defined as the biocatalysts which are produced by living cells. Sir, which is produced? It is living cells produced. So which are catalysis many chemical reactions. So it is act as catalyst. Catalyst means what? It is a positive catalyst increase the rate of the reaction. If it is a negative catalyst, decrease the rate of reaction. So in a reaction which is increased rate of reaction, which is decreased rate of reaction, that is we will call catalyst. So enzymes are act as catalyst. So based on that, it is classifies naming. It is classified as based on the naming. It is classified as based on the names. Based on the compound. Naming is there. Naming of enzymes. Naming of the enzymes based on what? Two types. Based on compound. Based on compounds or groups, based on the group of compound. Next, based on the reaction. Based on the reaction or reactions. I will tell what is that reactions. So first of all, this one based on the compound enzymes naming, how we will give. So hydrolysis of first one, hydrolysis of maltose. So hydrolysis of maltose means maltase formula C12H22O11 plus H2O. Hydrolysis no H2O we will get in the presence of enzyme. What enzyme? Maltase, ASE. So ASE is enzyme name ends with ASE. Maltase, we will get 2 moles of C6H12O6, that is glucose. 2 moles of glucose we are getting. What is the compound name? Based on the compound, what is the name? Maltose. Maltose converted into glucose by the help of maltase. Maltase is a enzyme now. So enzyme number 1. Second enzyme, hydrolysis of ammonia. Ammonia hydrolysis. What we are getting? Ammonia formula CH2 C double bond O, sorry, NH2 C double bond O NH2. Hydrolysis of urea, sorry, not ammonia. Hydrolysis of urea. So this is urea. On hydrolysis we will get ammonia, 2 moles of NH3 plus CO2 we are getting. CO2 and NH3. See, C and O, here 1 O, CO2. NH3 means 2 NH3, so 6 hydrogens, 2 plus 2, 4 plus 2, 6 hydrogens, 2 nitrogens. So it is balanced reaction. Here catalyst name only we want. What is that catalyst name? Urease. ASE, urease. So this is the catalyst. So this is also catalyst. So that catalyst what? Enzyme. That catalyst is enzyme. Based on the compound name only enzyme will come. Next, third one, hydrolysis of sucrose. CROUSE, hydrolysis of sucrose. So sucrose formula also C12H22O11, presence of water molecule, hydrolysis. Here we will write sucrase. Sucrose, no, SUCRO, here CRASE will come, sucrase. This is the catalyst name, sucrase. We will get 1 mole of glucose and fructose. Both are C6H12O6 only. C6H12O6, first one is glucose, second one is fructose. So 2 compounds will come, hydrolysis of sucrose. That is, one is glucose, second one is fructose. Here we are using one enzyme to cut this disaccharide. To breaks down this disaccharide, we will get one glucose and fructose with the help of sucrase enzyme. So maltase, urease and sucrase, based on the compound, this is the name of the enzymes. Now based on the reaction, some types is there. What is that reaction? If it is reaction is redox, means both oxidation and reduction involved. Oxidation, both reduction involved reactions is there, no, both present in one single reaction, that is redox reaction. At that time, enzyme we are using, means that enzyme name is reductase, oxidoreductase. This is the name of enzyme based on the reaction. Second reaction, oxidation reduction involved, means that one. Second one is transferase. This is also enzyme name. What is this reaction? Reaction means functional groups, already you know your 12th standard. Functional groups are transferred in a reaction with the help of transferase only it will happen. Transferase also one enzyme, oxidoreductase and transferase. Transferase, where it is we are using functional groups will be changes. This if second carbon functional group changes into one more carbon functional group, like changing, or completely functional group aldehyde from functional group changes into ketone, like we are using transferase enzyme for that. Next, third one is hydrolysis. If hydrolysis either remove, hydrolysis means what here? Hydrolysis only enzyme name, hydrolysis. This is enzyme. This is also enzyme. Transferase also enzyme. Oxidoreductase also enzyme. So hydrolysis to removal of or addition of what H2O, water, or H2O or ammonia, NH3, or CO2. These three addition or removal, we are using these catalyst. What is that catalyst? Hydrolase. HYDRA, hydra, LASES. These catalyst we are using either in a reaction we are adding water, or we are adding CO2, or we are adding NH3, or removal of these three also we are using this catalyst. This catalyst are this enzyme. This enzyme act as catalyst. Either increase the rate of the reaction or decrease the rate of reaction, we are using hydrolase, transferase, and oxidoreductase. This is the based on what the reactions. Next one more is there, I will write, no space, that's why, that is lyase, LY. Okay, sorry, I said wrong here. This hydrolase means reaction or enzyme brings, this enzymes bring hydrolysis of the compound. So hydrolysis, we are doing hydrolysis means we are using their enzyme name is hydrolase. So that one what enzymes that bring about hydrolysis of the reacting compound, hydrolysis of the compound. If we are doing hydrolysis means hydrolase we are using. Okay, that is that. Next, lysase, LYSASE, lysase. This is to remove, already explained, to remove H2O or addition of H2O or NH3 removing or NH3 addition, CO2 removing, and CO2 addition. So this one we are using one enzyme, that enzyme name is lysase. Okay, next one is isomerase. This is fourth one. Fifth one is isomerase. Isomerase means all isomeric compounds we should use increase rate of reaction, increases rate, we are using this isomerase enzyme. So this is also enzyme name, this is also enzyme name, this is also enzyme, this is also enzyme, this is also enzyme name based on the reactions. Next, last one, ligase is there. Ligase, sixth one. What is ligase? Ligase. Sixth one is ligase. It tells about synthetic reactions. Synthetic reactions. Ligo means in Greek word bind. Ligo means bind. So bind to, what is that? Two compounds. How to bind two compounds? So we want to add one more compound and one more compound, that binding this compound and this compound both are bind with the help of this enzyme. So synthetic reactions we are using this enzyme to bind the two compounds. So liga means bind. So ligase is the enzyme name. Enzyme names once again see based on the reactions. So oxidation reduction reactions we are using enzyme means that enzyme name is oxidoreductase. So transfer of functional group we want to do means transferase we are using. So hydrolysis we are doing means we are using hydrolase. Next we are doing removal of water and NH3CO2 and addition of NH2CO2 and H2O, we are using lysase. Next isomerase, we are using isomeric reactions breaks down, we are using isomerase. Next ligase means synthetic reactions, we are using ligase. Synthetic reactions, addition synthetic reactions. So liga means bind. This is about name of the enzymes. Now mechanism of enzymes mechanism. Enzymes mechanism. Enzymes mechanism. What is mechanism? So enzymes mechanism, what? This is one graph. This is reactant, here this is product. So this side we are taking energy, this is reaction coordinate. So activation energy about So with the help of enzymes increases in the reaction, enzyme adding, so activation energy will be decreases. That means what? Enzymes increase the rate of the reaction, energy of the reaction will be decreases. Energy are lowering the energy of the activation energy. Activation energy I will tell like this, activation energy, this word. So generally reactants changes into products. Here in this particular height of the energy, activation energy, this is without enzyme. Without enzyme, the reactant changes into product, this symbol. Okay, here this symbol. So here to here that height is without enzyme, this is energy, this much required. So this is we will call activation energy. This much energy required. So now with enzyme, what happened? With the enzyme we won't require that much of product also more came. See, energy is less. So this is this height we will call that height. So here to here, this height we will call, that height means here onwards I will take, see, this height here to here. Now this is what? With enzyme this height came. This is activation energy. With enzyme, activation energy without enzyme. Difference is there, no? So that means enzyme increases, activation energy will be decreased, this much decrease. So enzyme in the reaction, enzymes increases activation energy. energy energy decreases, increases. Enzyme increases, activation energy will be decreases. This is the reason, this is the mechanism of the enzymes. If enzymes is present, the reaction will be fruitful, it will come. So enzyme increases, activation energy decreases. The decreases is how much? See, this much decreases is this much difference is there. So activation energy this one. Okay, next two steps mechanism will be happen, that is step 1. Step 1, what happened now? Enzyme and substrate bind together form a one complex, enzyme and substrate complex. Next step 2, this decomposition of complex, binding complex. So that binding complex, how to decompose? ES gives less to E plus product will come. So we want product, for that we are adding enzyme to the substrate or reactant, it will get binding. After that binding one we are taking, and we have to use some enzymes or decomposes this one with the help of some temperature or energy, it will be changes into products. So this is the mechanism of enzymes. Next vitamins. So it is organic substance. Vitamins, it is a small organic substance required to a human being or animals or everything living organism, because without this one some diseases also will come. Extra is there also, some diseases will come. So and vitamins are mostly two types. So that is, one is water soluble, second one is fat or oil soluble. So water soluble we will call that is C vitamin and B vitamin or B complex. Next this one is ADEK. So these vitamins are fat soluble. So what about A means it is less or deficiency of A, eye problems will come. To rectify this, we should eat what? Milk and fish. Next D vitamin, rickets will come. This is disease, eye disease. Rickets means bones disease. To rectify that, here milk we are eating, milk and fish. Here for this one D vitamin, sunlight, rickets problem will come, sunlight and milk and fish liver, it is source. This is a source, this is a problem, disease. D vitamin source is this one. D vitamin is less, means we will get this disease. A vitamin source is this one, we will get deficiency, means we will get eye problems. Next E means what? E vitamin, it is present in sunflower, rich, it is RBC, red blood cells problem will come. So it is present in sunflower oil, K vitamin, leukemia, some K vitamin means blood anemia, hemophilia. Disease is hemophilia. So this is disease, it is green vegetables or green leaves we should take, means it is problem will solve. That is that. Now here water soluble C vitamins, this is present in citrus fruits. It is diseases of this one is C vitamin, C vitamin present in some C vitamin diseases, skin problems. This is disease. We are eating C vitamin fruits are amla, ascorbic acid, amla fruits or citrus fruits, that and all we should eat means okay. Now B complex have some names is there, amma, that is thiamine means B1, vitamin B1, thiamine. Next riboflavin, second one is riboflavin B2, vitamin B2. Third one is riboflavin after niacin B3, Fourth one is B vitamin B6 pyridoxine B6. B5 is pentanoic acid, pentathionic acid. So this is B5. Okay, next B12 is there. B12 is cyanocobalamin. Cyanocobalamin. If it is less is there, means what problem will come? Blood leukemia, na blood disease. Folic acid B9, folic acid. Sixth, this is seventh amount, folic acid we will call B9. So this is about vitamins. So thank you, subscribe my channel once again. Thank you everyone. So this is the 12th standard biomolecule chapter. So next class we will discuss about one more concept, that is nucleic acids. Thank you.